In this video, we're going to talk about the specifics about the DNA replication process with all the different enzymes involved. So we're going to explain the process of DNA replication. I know it says in prokaryotes, but just think of this in general. I'm not sure why I'd be specific about prokaryotes. And we're going to talk about a bunch of different enzymes. So helicase has a special job. There's two different types of polymerases that we're going to talk about. RNA primase, what does it do? DNA gyrase, these binding proteins, which really aren't enzymes, ligase, and then these Okazaki fragments that are created, and the role of this type of, excuse me, this type of nucleotide. So I'm going to use this slide just to explain it because they did a nice job here, I think, as far as the image, and we'll just use it. So the first enzyme I want to talk about is not in the picture. After I said so many great things about the image, the first enzyme is not in the picture, and this is gyrase. So gyrase's role is to release the stress. It's kind of loosening the double helix. So if you remember, the DNA double helix is so tightly wound in this coil. So its job is to loosen it. Also, up here are little proteins. I've seen them drawn them as like little balls, like before and after the gyrase. But there's proteins here, these binding proteins that kind of stabilize the whole structure. So that's what this does. Gyrase kind of loosens the stress, and these binding proteins are there just to stabilize the whole replication process. All right, so let's talk about replication. So we talked about stabilizing and unwinding. The first enzyme to come through is this enzyme called helicase. What helicase does is it moves straight through here, and it breaks the hydrogen bonds between our bases. So adenine and thymines, 2 hydrogen bonds are broken. Cytosine and guanines, 3 hydrogen bonds are broken. So they're broken. Once they're broken, they've exposed, right? These bases that were once connected with their partner are now just kind of like sitting out in the open. So, so for example, if this one was adenine, then if this one was thymine, it could float through the nucleus and eventually find its base pair, adenine, and it would form a hydrogen bond there. And maybe the next one is cytosine, so guanine would come over. And maybe the next one is adenine, and so thymine would come over again. Maybe the next one is guanine, and maybe a cytosine would come over. So it forms those pairs. I'm just going to try to fix this real quick because I wanted to demonstrate something. So I need to. All right. So we have our free nucleotides forming complementary base pairs to their partner. Now, I purposely left spaces between them. That's because, as we were talking in the previous video, that the phosphate from one nucleotide has not joined carbon number 3 to the next one. So, right? So we have a phosphate, our deoxyribose, and then our nitrogenous base. This needs to form a bond with the one above it. I'm writing in the text, it's horrible, but anyway. So this has to form, and that's why we don't have a line here, because this bond has not formed yet. And that's the role of this enzyme. DNA polymerase 3, its job is to connect one nucleotide to the next by creating covalent bonds between the phosphate here and then carbon number 3 of our other nucleotide. Next, the next enzyme I want to talk about is this RNA primase. So RNA primase is an enzyme that creates this thing called an RNA primer. So primer means like to start. So what it's done is it's identified with this red set of nucleotides it's identified where the replication is going to begin. And yes, it's RNA. So this could actually be a uracil, not a thymine. Very possible. So it identifies where the replication process starts, and then the replication process begins from our 5' direction, right, to our 3' direction, just like we talked about in the previous video. Okay, so that's how it works on this strand. This strand is simple. It's called the leading strand because it just goes in a simple one-way direction. As we keep breaking these bonds, it will keep going in this direction. However, on the other strand, which is called the lagging strand, it's a bit of an issue because our 5' is here on the new strand and the 3' is down here. So we have to copy in this direction because that's the law in DNA replication. So things happen a little differently here. So one thing that's still the same is that we have this RNA primer here created by the RNA primase. But what happens is, as we open this up, as we unzip the DNA, we will drop another primer behind it and then copy in this direction. And as we open up more, we will drop another primer behind it and we'll copy in this direction. And what happens is, between the places where you started replication, you get these little holes, like these little spaces in your DNA. Because we started here and we went in this direction. We started here and we went in this direction. So we have this little hole here. So those little spaces, that we fix them with an enzyme called DNA ligase. And what it does is it joins these fragments. Should use a different color than blue. It joins these fragments together, which are called Okazaki fragments. So these little pieces of DNA are called Okazaki fragments, the ones in between, because we're kind of going backwards. I mean, we're still going 5' to 3', but as we move this way, we're going to go backwards. And the last enzyme I want to talk about is this enzyme called DNA polymerase 1. And DNA polymerase's one job is to take all of these primers and convert them back to DNA, because we don't want to have RNA in our replicated DNA. Okay, there's one part we didn't talk about that I wanted to mention. So they want you to know the role of deoxynucleoside triphosphate. So I'm going to show that on the next slide, just so we can see it better. So I'm going to draw, on the right-hand side, I'm going to draw a nucleotide. I'm going to have it pointed down this way because the two are anti-parallel. Here's my phosphate connected to carbon number 3 on the next nucleotide. It's connected to a nitrogenous base. Not very good. And then to a phosphate. So here are two nucleotides joined together on an anti-parallel strand. The new pieces of DNA, I'm going to represent them with blue. So here's a nitrogenous base connected to deoxyribose, and then a phosphate coming off here. And the next one, the next new piece, the next new nucleotide, right, in this replication is going to come here. Because if you remember, we always go 5', here's my carbon number 5, to 3', carbon number 3. So the new one's going to connect down here. When the new one comes over, it has 3 phosphates, hence the name triphosphate. I'm going to draw another one up here. And it's connected to its sugar, connected to a base. Wow, this is some really bad art. So not connected, right? We're not connected here. In order for this to form that covalent bond here with the help of DNA polymerase 3, it needs the energy in the bonds, kind of like ATP, adenosine triphosphate. This is nucleoside, deoxynucleoside triphosphate. It needs the energy in the bond between phosphate number 1 and phosphate number 2. So this energy will allow the covalent bond to form, and then these two phosphates are recycled back into the cell. So that's the role of deoxynucleoside triphosphate, providing the energy to form those covalent bonds between the phosphate of one and carbon 3 of the other. And the last thing I want to talk about, and it'll be brief, IB wants you to know that DNA replication happens at many locations on a molecule of DNA. So here it says, like, if we just went from the beginning and all the way to the end in just a sequence, it would take 17 days to create just one chromosome. Not efficient. So what it does is it opens up, helicase will come in many places to open up that DNA and start replication in multiple locations. So here's my double helix, and wow, that's really not very good. So maybe my helicase started here and it's replicating this way, so we opened it up here, and maybe there's another helicase here, opening up our DNA and copying it this way. This image is pretty good because you can see like 1, 2, 3 locations on this chromosome where the strands have separated so they can make copies of their DNA.